So for lecture tonight, we're going to get through the parts A and B in chapter 3, the first two PowerPoints. However, the first part in chapter 3, part A is a review. So I'm just going to go over it rather quickly. Chapter 3 is all about cells and how things are transported across cells. So I'm going to kind of skim over these first few slides that go through cells, the living units. They talk about cell theory, cell diversity, explain some of the different types of cells. And again, I know I'm going through this rather quickly, but I just want to get through the bulk of it into cellular transport. And then it kind of takes you through the general structure of a cell, the extracellular materials, the plasma membrane, and what makes up the plasma membrane of a cell. You should understand how membrane lipids form a lipid bilayer in the membrane. And I just want you guys to have a good understanding of the plasma membrane and some of the proteins that can be within that plasma membrane. And again, I'm going through it quickly because we would normally have like an hour-and-a-half lecture, but we're doing kind of this hybrid learning environment. So that's why this is kind of a combination of online learning and me lecturing. The phospholipids form the membrane structure itself, and then you have different proteins that are embedded within the membrane to help with cellular communication and some sort of reception of what's going on extracellularly to tell the cell what to do in a situation. So you have different membrane proteins and then different ways that proteins will transport things into and out of the cell. Called transport proteins. These PowerPoints have a lot of great animations as well. You just have to download them to your computer to actually see them all. I'm just going to skip ahead here to where I wanted to start with cellular transport, which is around passive membrane transport. Okay, so here we're going to start talking about passive membrane transport. Passive and active transport has to do with how cells get substances into them and how they get substances out of them. And this is really important because cells need to take up things that are exterior to them to be able to perform their function. And this will be really important when we talk about physiological processes, especially with the nervous system. I start with passive transport because right here, I'm going to go back to your Canvas really quickly because I want to record this as well. One major thing that you guys are getting also graded on are physiology workshops. And I posted a recent announcement about this. You guys will get graded on 3 physiology workshops, and they're due by the end of the semester. To get all the information about physiology workshops, you can go to this page in the announcement, and it takes you through how to complete physiology workshops. You check the live session schedule, and then you'll attend a Zoom workshop meeting. It's over and above what we do in class, but you get 30 points for attending 3 of them, 10 points each. You fill out the handout, and then you email it to the MDSC Center, which is right here. So I wanted to bring this to your attention. Here are the online lectures and the handouts for each of the workshops. And then you can go to the schedule, and I'd encourage you guys to check out the schedule right away. Can you guys see the schedule on your screens?
Yeah.
Good. Okay, so this is the physiology live schedule. I teach some of these as well. You can see I'm signed in there. And this tells you the topic of the workshop itself and then a time that you can attend a workshop on that topic. The MDSC is the Multidisciplinary Success Center, where you can also look up tutoring. And they do these workshops to line up with when you guys are having tests on the topic. So try to attend a workshop before you have a test on it. And that's why, let's see here. I focused tonight's lecture a lot on things that will have to do with graded and action potentials, but I just wanted to, any questions on physiology workshops?
I do. Are they all the same?
Yeah, so all the ones on graded and action potential, the 3 instructors use the same material, you do the same handout, you watch the same introduction video for them. They take
Okay.
about 45 minutes.
All right, thank you.
Any other questions? So you have to attend only 3 of them. I would encourage you guys to attend all of them because they're all on topics that are really difficult to understand in physiology. So they're like an extra supplemental learning for some of these more difficult physiological topics. Any other questions? Yep, you can choose any 5 that you want, any 5 to get credit for. So you fill out, well, you fill out only 3 to get credit for 3 that you would fill out a handout for. But go to all 5 of them if you can and just listen in. Okay, the handouts are fillable. So it's like a Word document that you just type in answers and then you send it on to the MDSC, this email address. Do you want me to show you an example of one of those handouts? Can you still see my screen? So this is a handout for the cardiac cycle, for example. And if you just download it and let me know if can you see my Word document still that came up? Anyone, do you guys see the Word document that came up? Okay, thanks.
No.
No, you don't. Okay, yeah, I figured when I open up a new thing, it leaves you there. Okay, here we go. So now you should see it. So this is an example of the handout for the cardiac cycle. You'll fill out your name, my name, Elizabeth Bamer, the course number, what are we, Physio 1, and then it gives you the instructions again. You can watch the workshop video, a link to it there. You'll probably recognize the voice in those workshop videos. I made those this summer. Then you'll take a link to a heart sound animation. So again, it's just supplemental material. Then you'll fill out the handout. You can usually fill out the handouts when you watch the workshop videos, but when you attend a live workshop instruction, and again, they're like 45 minutes long, they're taught by one of the instructors, me, another gal who has her PhD in physiology, and then another guy who's taught physiology for many semesters. So they can help you with some of these questions, and they might take you through some sort of activity. Other questions about that? I'd probably wait till the end of the semester until I just get all of them. Yeah, so you can verify with me that I've received them. And when I receive them, I can put them into Canvas that I've gotten them. I'm not sure if the MDSC gives them to me all at once, or if they give them to me as you complete them. But if they give them to me when you complete them, I'll submit your grades, like 10 points at a time. All right.
I have one more question. Do we need to submit it before or after?
So yeah, they don't really, so yeah, you probably should submit it after you attend the live instruction part of it. But to be honest, they won't check because they don't really take attendance in those live workshops. It's just an honor system. So you can submit those handouts whenever.
Great, thank you.
If you submit all 3 at once, they might get a little suspicious, but yeah, you only have to complete 3 handouts. You have the opportunity to do all 5.
Mm-hmm.
Okay. Let's see, other questions before we get started. It's a great question, Esmeralda. Let me get back to you on that. I would potentially give you extra credit for doing all 5, yes. But you would have to do all 5, not just, I won't give you credit for doing 4 of them. I would give you extra credit if you do all 5 of them. Okay. If you have any other questions, put them in the chat. Otherwise, we'll start to get started here. And I start with passive transport. And then part B of chapter 3 goes through active membrane transport. And this is what I'm going to focus kind of my lecture on. And then on Thursday, we'll get through parts C and D. So when we're talking about passive and active transport, we're talking about things coming into the cell and leaving the cell and going through the plasma membrane. And there's 3 types of passive membrane transport. And when it says passive, that means it requires no energy input. So these things will happen, I think they'll happen naturally. They'll happen without ATP. And there's 3 main types of passive transport: simple diffusion, facilitated diffusion, and osmosis. And all of them involve diffusion, which is what we call the natural movement of molecules from an area of high concentration to low concentration. And we refer to that as the concentration gradient. All molecules have random high-speed movements where they're bouncing off of each other. Movements will result in a collision between the molecules, and molecules in higher concentration will collide more, resulting in them to be spread out or scattered to lower concentration. So a couple examples of this is if you were to spray some perfume in the room, that perfume where it's initially sprayed would be highly concentrated. Eventually, it will diffuse throughout all parts of the room until every area in the room has an equal concentration of that perfume. So that's just simple diffusion, moving from an area of high concentration to low concentration. Here you see an example of a dye pellet, and eventually over time, the dye pellet will diffuse into all areas of the beaker. So that's simple diffusion until all areas of the beaker are equally concentrated with that. The speed of diffusion is influenced by the concentration gradient, meaning the greater the difference of concentration between the two areas, the faster the diffusion occurs. And that makes sense. If you spray a bunch of perfume in the room, it will diffuse faster and get to all areas of the room faster. Molecular size, smaller molecules will actually diffuse faster. And temperature, higher temperatures increase the kinetic energy between molecules, so they'll just bounce off of each other more, and that'll help to increase diffusion. Equilibrium will be reached when there's no net movement of molecules in one direction only. So this is diffusion. More about diffusion. It's called simple diffusion because molecules that are able to passively diffuse through the cell membrane include anything lipid soluble and anything non-polar. And that's because the cell membrane itself, the inside of it, is made of lipids, those lipid tails from the phospholipid bilayer. So molecules will passively diffuse through the membrane, and this is called simple diffusion. Again, diffusion happens without any energy. Larger or anything non-lipid soluble or polar can also cross through the membrane, but with the assistance of what we call a carrier protein or a carrier molecule, and we call that facilitated diffusion. Facilitated diffusion still happens naturally down its concentration gradient, but there's usually some sort of carrier molecule to help the substance get across the cell membrane. Osmosis then is the third type of passive transport, and it's a special name for the movement of solvent, most often water, not molecules. This is important if a plasma membrane is severely damaged. Substances could diffuse freely into and out of the cell, and that could compromise the concentration gradients and compromise the cell itself. And this is common in burn patients. They can lose precious fluids, proteins, and ions. We say that they weep from the damaged cells because they are getting out of the damaged cells more quickly because their cells are damaged. So common in burn patients to weep fluids out of their cells. Simple diffusion, an example in your body of things that go through simple diffusion are when oxygen and carbon dioxide will diffuse across your lung air sacs to get into your blood. And we'll go into that more, but again, these introductory chapters kind of give you an introduction to some of these physiological processes that we'll go into more detail later. Steroid hormones, fatty acids will travel across cells through simple diffusion. This is an animation of diffusion that, again, I'd encourage you to watch on your own when you download the PowerPoint. And this shows an act of simple diffusion traveling across the cell membrane. And again, the cell membrane is just the exterior barrier of the cell, showing how there's a higher concentration of molecules outside the cell membrane and how they will diffuse the cell membrane through the cell membrane into an area of lower concentrated. So this is simple diffusion. Facilitated diffusion, again, this is if anything hydrophobic or too large, like glucose, amino acids, or ions, they cannot get through the cell membrane on their own, but they will still travel down their concentration gradient. So they'll use some sort of facilitator or carrier protein. And it can be a carrier-mediated facilitated diffusion, which are substances will bind to protein carriers themselves, or it can be channel-mediated, where substances will move through a water-filled channel. It gives you more detail, maybe more than you need to know about carrier-mediated facilitated diffusion, showing how some sort of lipid insoluble solute will travel through the cell membrane using the help of some sort of carrier. The one will go in at a time, and then it'll spit one out at a time. So this is carrier-mediated facilitated diffusion. It does not require energy. It happens on its own. It'll help things from going from an area of high concentration to low concentration via passive transport. Then channel-mediated facilitated diffusion just uses a channel, which is an aqueous-filled core, meaning it has a core filled with water. Water channels will be called aquaporins. There's two types of channels. They can be leakage channels or gated channels. Leakage channels are always open. They'll leak things through the cell membrane. And gated channels are controlled by either a chemical or electrical signal. And we'll talk about gated channels when we get more to your nervous system. So here's just a look at a channel-mediated facilitated diffusion. And again, things will diffuse through a channel protein. Mostly ions will be selected on the basis of their size and charge. So this is facilitated diffusion. And then osmosis is our third type of passive transport. And osmosis is the movement of water across a selectively permeable membrane. Water is able to diffuse directly through the plasma membrane, through the lipid bilayer, through specific water channels called aquaporins. It will occur when water or another solvent concentration is different on the two sides of the cell membrane. So what this means, this is just showing in osmosis, water is the one that is diffusing through the cell membrane. And all the other types of transport, we've talked about a solute diffusing, whether that's sugar, an amino acid, something too large to get through the plasma membrane on its own. But osmosis is only about water movement, so water diffusing through what we call a semi-permeable membrane. And the cell membrane is semi-permeable, meaning it only allows certain things through it. Osmolarity measures the concentration of the total number of solute particles in a solvent. You don't need to understand too much about osmolarity. It has to do with water concentration relative to the number of solute particles within it. If you've taken chemistry yet, you've probably heard osmolarity before. Water will always move by osmosis from areas of low solute concentration to areas of high solute concentration. What this means is that water just always will move into an area of higher solute concentration. It'll continue to move until equilibrium is established on both sides of that semi-permeable membrane, where there will be a same concentration of solutes and water molecules on both sides. Okay, so the best way to kind of talk about osmosis is to see a visual. Here is a look at a U-shaped beaker showing how we have a freely permeable membrane to begin with. So this is an example of a freely permeable membrane, meaning that both water and the solute can move through the membrane itself. So simple osmosis will occur. Water will move both ways, and actually the solute will move from an area of high solute concentration to low solute concentration, and water will move into an area of higher concentration until equilibrium is reached and you have equal levels of water and solute on both sides of the membrane. So this is a look at a membrane permeable to both solutes and water. Osmosis and diffusion will both occur. Osmosis occurs because water moves into an area of higher solute concentration, and diffusion occurs because the solute wants to spread out until equilibrium is reached. Osmosis will also occur when the membrane is semi-permeable, meaning that solutes are unable to pass through the membrane, only water is. And now we're going to look at how osmosis occurs within a selectively permeable membrane. So you can see here how the solute molecules are unable to pass through the membrane because they're too large to get through that membrane. And again, this all relates back to cells and their membranes and what is able to get through and how water can get through. So in this situation, water is the only thing that can get through this membrane. And water will want to move into an area where there's more concentrated solute particles. And when water does that, there will be more water on the right side of this beaker, and that will raise the water level. So here at the end of the osmosis, we still get equal concentration because we have an equal ratio of solute particles to water, and there's more water on the right side of the membrane to equalize those solute concentration levels. A great way to understand this too is to remember the rule that solutes suck. Solutes will always suck water into an area of higher solute concentration. So that's the way that water wants to travel, from an area of high water concentration to low water concentration. And that's why at the end of this equilibrium process, there will be more water on the right side of the membrane because there's more solute particles on the right side of the membrane. Does that make sense to everybody? How water travels into an area?
Yes.
Really? Okay. I know this is a lot of explanation about this, but I just wanted us to be clear. I usually ask questions about what I explain more on. The movement of water involves hydrostatic pressure and osmotic pressure. Hydrostatic pressure is outward pressure exerted on a cell, often called back pressure, and osmotic pressure is inward pressure due to the tendency of water to be pulled into a cell with higher osmolarities. And again, an osmolarity, higher osmolarities refer to the concentration of solute. This takes you through a little more in depth on hydrostatic pressure dealing with plant and animal cells. Plant cells are surrounded by a really strong cell wall that help to limit hydrostatic pressure. So plant cells actually only fill with so much water and then they'll just stop. Animal cells do not have cell walls, therefore they cannot limit hydrostatic pressure. So animal cells can burst if too much water is taken in, and these animal cells can also shrink if water will leave the cell. And that's where we get into tonicity. And this is the ability of a solution to change its shape or tone of its cells by altering the cell's internal water volume. And we'll talk about this as it relates to red blood cells. And it's really important for our red blood cells to be in an isotonic solution. And this will be important when you administer a saline solution to someone to make sure it's the right tonicity or concentration of solute so that your red blood cells will be happy in it. So 3 types of solution. We have isotonic solutions, hypertonic solutions, and hypotonic solutions. And the solution always refers to the concentration of an area or a solution outside a cell. So an isotonic solution would have the same osmolarity or concentration. It's not actually concentration, but for our purposes, I'm going to use those 2 words interchangeably to help understand it or explain it. An isotonic solution has the same osmolarity as inside the cell as outside, so the volume will remain unchanged. A hypertonic solution has a higher osmolarity or concentration of the solution itself than inside the cell, so water will want to flow out of the cell, resulting in the cell actually shrinking when placed in a hypertonic solution. And we call that shrinking crenation. And I'll show you examples of this when we get to those red blood cell pictures. A hypotonic solution has a lower osmolarity or concentration than inside the cell, so water will flow into the cell itself, resulting in the cell to swell and possibly bursting. So if your cells, red blood cells, are placed in a hypotonic solution, water will flow into the cells, causing them to possibly burst or lyse. This gives you an example of sodium chloride and showing in a moles. I'm just going to pass over this for now. We're not going to get into a lot of chemistry questions. Well, I would love to. I was a chemistry major. I tell people that all the time to make you think I'm smart, but I'm really not. Okay, so this is an example of placing your cell. If we place a red blood cell, so these are red blood cells, they don't have nuclei. They lack a nucleus mainly to hold as much oxygen as possible in them. If we put a red blood cell in a beaker of an isotonic solution, that means the solution outside of the red blood cell is the exact same concentration as inside the red blood cell. So water will osmose in and out of the red blood cell, but not in any net direction. So the red blood cells are happy because the concentration outside the red blood cell is the same as the concentration inside the red blood cell. If we put a red blood cell into a hypertonic solution, that means surrounding this red blood cell right now in black is a hypertonic solution. That means the solution is more concentrated with solute than what is inside the red blood cell. This is bad because water will naturally osmose passively. Remember, this is a passive transport process, so no energy is required to do this. Water will just naturally flow out of the cell into that highly concentrated hypertonic solution. When that happens, water leaves the cell and it will shrivel up, and we call that crenation when the cells will shrivel up. So not good. If we put your cells, red blood cells, into a hypotonic solution, we're putting them into a solution that's less concentrated than what's inside of them. So water, again, always travels into an area, solute suck, of more concentrated inside the red blood cell. So when water travels inside the red blood cell will swell, possibly swell so much like a balloon that it will actually burst or lyse, and that is called cytolysis, cell splitting. So what happens if you are marooned at sea and you're so thirsty and you drink salt water? Why is drinking salt water bad for your cells? What do you guys think?
Because it has sodium, like
Yes.
it's so
Dries out the cell.
It dries out your cells. So yes, it has sodium chloride in it. It'll dehydrate your cells. When you drink salt water, you're basically drinking hypertonic solution. You're drinking a ton of hypertonic solution. You're covering your red blood cells, all of your cells really, not just red blood cells, in this really densely concentrated salt water. And naturally, all of your cells, to try to maintain homeostasis, water will leave all of your cells to try to equalize the concentration of that salt water that they're being placed in and the inside of the cell themselves. So all of your cells will shrivel up, and that's what dehydration is. Your cells are losing water. Also, if you guys are fishermen, it's important that you never put a saltwater fish in a freshwater tank or vice versa, because the same kind of thing happens with your fish cells. They also have other mechanisms to help balance their salt water, but the same type of thing happens with fish as well. Okay, perfect. It's 7 o'clock. We're going to start the next PowerPoint slide. Any questions on passive transport? Thanks, Max. No questions. All right, let me just exit out this and I'll pull up part B. And again, I know this is going fast through here. So I'm really encouraging you guys to be independent learners in some of this stuff to cover the first half of that PowerPoint and anything that you might have questions on. Okay, can everyone see my chapter 3, part B slide? Seeing no objections, I'm assuming you guys can see this. So part B starts in active membrane transport. So there is reason for why I'm separated out like this, because we're basically going through passive and active transport tonight. Active membrane transport. So this is getting things into or out of the cell, but they're all active transport processes. And this means that they both require, they can be active transport or vesicular transport. And they both require ATP to move solutes across the plasma membrane. ATP is your body's energy currency. So think of it, it's the money in your body that every process usually needs to perform a function. It needs ATP. So some things are too large for channels. They're not lipid soluble, so they can't get through the plasma membrane on their own, or they're not able to move down a concentration gradient. So if you need to get a solute particle across the plasma membrane, but there's too much of it outside the cell, it would be like trying to force your way into a really crowded subway car or a train. You need energy to force your way into a bunch of people. So these require active transport processes. They require ATP. Some of them will require carrier proteins, and we call those pumps. Some carriers transport more than one substance at a time. We call them antiporters. If they're going in opposite directions, one into, one out of the cell. One are symporters, if it's a pump that transports two different substances in the same direction. They'll all move solutes against their concentration gradient, meaning active transport processes will always move something from an area where it's lowly concentrated to an area that's more highly concentrated. And again, think of yourself trying to push yourself into a subway car, into a crowded concert arena, even though we're probably not going to concerts anytime soon. So this will require more energy to do that. Two types of active transport. They can be primary active transport, where the required energy comes directly from ATP hydrolysis. That means ATP is a molecule, and when we break it apart, energy comes out to fund these active transport processes. Secondary active transport, the required energy is obtained indirectly from ionic gradients created by some sort of primary active transport. Primary active transport, this happens when we break apart ATP. That causes change in shape of some sort of transport protein. An example of primary active transport is your sodium-potassium pump. And this is in bold for a reason. The sodium-potassium pump, we're going to come back to this many, many times throughout the semester. Sodium-potassium pump is an active transport process. It requires ATP to function. If you learn anything tonight, sodium-potassium pump is an active transport process. It's also our most studied pump because it's in your nervous system. It's all throughout the body. It's basically an enzyme called sodium-potassium ATPase. It pumps sodium out of the cell and pumps potassium into the cell. So again, it's an antiport because it'll pump one thing out and another thing in. Pumps sodium out of the cell, potassium into the cell. It's located in all plasma membranes, so that's why it's the most highly studied, it's the most known, but it's especially active in all sort of excitable cells, like your nerve cells and your muscle cells. Leakage channels located in membranes result in leaking of sodium into the cell and leaking of potassium out of the cell. So sodium-potassium pump just works to reestablish concentration gradients. It's important to have a certain amount of sodium outside your cell and a certain amount of potassium inside of the cell for all of your nerve cells to function. So that's why this pump is incredibly important, to establish this concentration gradient so that all of your nerve impulses can happen the way they should. And this is what it talks about by maintaining an electrochemical gradient, which involves both concentration and electrical charge in your nervous system. To send out an electrical signal throughout your nerves, it's all about changing the electrical charge of axons in your neurons. And this electrochemical gradient is maintained by your sodium-potassium pump. Here's the primary active transport process. This is just showing the sodium-potassium pump, showing how ATP will bind and then be hydrolyzed or broken apart to get sodium into the cell and to get potassium out of the cell. And this will take you through the steps of that sodium-potassium pump, and I'll let you guys kind of work through those steps on your own. I probably won't ask you too many details about each individual step, just knowing the general overall process of the sodium-potassium pump is important. Resting membrane potential, this is I'd encourage you guys again to watch all these animations. Resting membrane potential refers to the electrical charge on your nerve cells when they're at rest, meaning when there isn't a electrical stimulus being pumped through your nerve cells. secondary active transport was the second type of active transport. This will depend on ion gradient that was created by primary active transport. Usually energy stored in these gradients is used to indirectly drive. So a secondary active transport uses the pump, but in an indirect way. And I'll let you guys look through this. Here's primary active transport versus secondary active transport. Sodium glucose will be co-transported in and will release glucose into the cytoplasm. So sodium will be transported in using the sodium potassium pump, but glucose goes along with it. So it's a way to get glucose into the cell by a secondary mechanism by using the sodium ions. When I say sodium, it's always Na. I'm sorry about that. I probably spoke too fast. And potassium is always K. And they have a positive charge to them because they've lost an electron. So they're called an ion. Remember, an ion is anything that loses or gains an electron. So that's why they have a positive charge on them. Okay, vesicular transport also requires ATP. And vesicular transport involves the transport of large particles, which are also called macromolecules, and also fluids across the membrane in membranous sacs called vesicles. So a vesicle is just like a bag or a sac that takes things into and out of the cell. So we call it vesicular transport. Vesicular transport processes include endocytosis. Endo means inside. So endocytosis transports things into the cell, and there's different types of endocytosis that we'll go over. Exocytosis is transport out of the cell, and there's vesicular trafficking where they can transport from one area or organelle in a cell to another. So endocytosis usually involves the formation of a vesicle. It usually also involves receptors, so it can be very receptive for what it allows to come, very selective for what it allows into the cell. The substance being pulled in must be able to bind to its unique receptor. Some pathogens can hijack this receptor to try to get into your cell to cause some sort of dysfunction or disease. Once the vesicle is pulled into the cell, it can fuse with a lysosome, where the lysosome would help digest whatever it is, or it can undergo transcytosis, so it can change. So really simply, endocytosis, the substance will fuse with a receptor in the cell membrane, and a vesicle will be kind of pinched off of that cell membrane and formed that way to get into the cell. From here, it will go to a lysosome to try to be broken down more, or it can fuse with some other sort of transport vehicle to try to get whatever it needed, which is usually a protein, into the cell. So that's endocytosis. And this takes you through the steps individually that, again, you can read through on your own. Phagocytosis and pinocytosis. Phagocytosis means cell eating. So this is when your cells need to take in something that's larger. It's usually some sort of solid particle that needs to be engulfed. A vesicle is formed around it, and this is phagocytosis, cell eating. And this is phagocytosis, anything larger like a solid particle that needs to get into the cell. This is important. So why do your cells need to do this? If your cells need to digest something or get rid of anything harmful in your body, they will basically engulf it by phagocytosis, and then your lysosomes will digest it. So if you think about big picture, this is a really important in your body to be able to engulf anything large to try to get rid of it. Pinocytosis is cell drinking. If you're wine drinkers, what is it? The Pinot Grigio? I don't know, it's some sort of Latin roots for pinocytosis is cell drinking. So this is when the cell needs to take up anything that's fluid. So it takes it up via a similar mechanism. The fluid particles come into the vesicle and get taken into the cell. But pinocytosis is cell drinking, so anything smaller. Questions? Okay, I'm gonna keep going just on a scale of 1 to 10, write in the chat how confused you are. 10, what should 10 be? 10 be, I feel good about it, I understand it. One being I'm really lost still, and I have no idea what we're talking about. And I'll keep lecturing, but you guys can, and then I'll look at the chat. Again, that's a quick way for me to see if you have a blank stare on your face. All right, vesicular transport, receptor-mediated endocytosis. This just talks about how there's receptors lining the pits that take in the substances, and they are able to be very selective in what they take in. So this is receptor-mediated endocytosis. Okay, not many things, less a couple. Okay, thanks. Exocytosis then is the process where material is ejected from the cell. It's usually activated by some sort of cell surface signal or changes in membrane voltage. So anything being ejected will be enclosed in a vesicle, and it kind of is spit out of the cell, kind of like vomiting. Substances that are often exocytosed or given up are hormones, like a neurotransmitter, mucus, and any sort of cellular waste. So these are things that are exocytosed. Hormones is good. If hormones are being made in a cell, they'll get exocytosed. A neurotransmitter will be exocytosed from a cell to continue some sort of electrical signaling process to a muscle, mucus, and all other wastes. So this is what exocytosis looks like. Whatever's inside the vesicle fuses with the membrane, and then the cell membrane will open up and basically spit out or vomit what was inside the cell. And I just think this is such a cool picture down here. It's actually showing under 100,000 times magnified of a cell exocytosing its contents in a vesicle. Kind of cool. And I'll let you guys read through this. This takes you a little bit more into the biochemistry of it, which specific plasma membrane that it will be bound to. And so each of the vesicle will have a receptor to it, which will bind to a specific plasma membrane receptor. So everything is very coordinated, specific to it. Okay. Oh, that's the cool picture that I wanted to show. Isn't that crazy, guys? Isn't biology so cool? I know. All right. Resting membrane potential then. This is the electrical potential energy produced by the separation of oppositely charged particles across the plasma membrane in all cells. So this is important because your cell membrane has a voltage to it, meaning it's charged. And this is especially important in your nerve cells and your muscle cells. This voltage occurs only at the membrane surface, and at the rest of the cell and the extracellular fluid are always neutral. So there's only a voltage at the membrane surface. Your membrane voltages range from -50 to -100 millivolts. So that just means they have a charge to them. The negative sign indicates the inside of the cell is more negative to the outside of the cell. Potassium is K, and it's a key player in maintaining a normal resting membrane potential. So potassium is positively charged, and it will diffuse out of the cell through leakage channels. It'll be pulled back in through the sodium-potassium pump. And again, your cells try to maintain this resting membrane potential. And that's important so that your cells are ready to be activated in some sort of nervous system response. So most cells have a resting membrane potential around -90. And the electrochemical gradient of potassium, this positively charged ion, sets the resting membrane potential. We'll really get into this when we get to your nervous system. But again, it's just important to know that on the cell membrane, there's a charge. So your cells, their membranes are electrically charged, and we call that charge the resting membrane potential at normal, kind of neutral homeostasis. And potassium and sodium, potassium ions, will maintain that resting membrane potential. Sodium can also affect the resting membrane potential. It will be attracted to the inside of the cell because of the negative charge. If sodium enters the cell, it can bring the resting membrane potential up to -70. Again, we'll really get into this, and this is why you should attend all those workshops, because they take you through some of these harder concepts. But again, because we're talking about just about transport processes of the cell, and we talked about the sodium-potassium pump, that's why we're introducing this idea of a resting membrane potential, which is again just showing you that The inside of the cell is usually charged around -90, and it maintains that charge of -90 because of these big protein anions. The inside of the cell will always be very, very negative because of these protein anions with a negative charge. So the inside of the cell is always very negative, but these positively charged potassium will move in to maintain the inside of the cell about -90. This just shows you how the potassium will either diffuse in and out to maintain the membrane, the resting membrane potential around -90. It's maintained through the action of your sodium-potassium pump. And again, your sodium-potassium pump is an active process. It will eject 3 sodium out of the cell and bring 2 potassium into the cell simultaneously. All right, other cell environment interactions. Let's see, how are we doing so far? We just have 12 slides to get through and then we'll be done here. What we just went over previously, that's really important, what I want you guys to focus on. What we go over right now is kind of supplemental material that'll help you understand some of the physiological processes as we get to them in the semester. Cells can interact with their environment by responding directly to other cells, and these interactions always involve glycocalyx, which is some sort of sugar protein. We call them cell adhesion molecules, CAMs, or plasma membrane receptors. And this is again how cells interact with their environment. Every cell has thousands of sticky glycoprotein CAMs projecting from their membrane. And again, they just function to do some sort of these that are listed here. They'll interact with other cells, they'll form some sort of matrix, they'll connect to each other, they'll help to direct cell migration, proliferation. These receptors will be important in contact signaling, chemical signaling, which will be an interaction between a receptor and some sort of ligand, which is a chemical messenger. So all of this kind of this interaction between your cells and the environment require all of these proteins to help signal them for what they should do. Chemical signaling will be important, also called ligand signaling, because when any sort of chemical or ligand will bind to it, the protein will change shape and cause the inside of the cell to change. We also have what we call G protein linked receptors, which is just an indirect way to cause a cellular change on the inside by activating G proteins. G proteins can cause a signaling change on the inside of the cell by releasing internal second messengers, which are called cyclic AMP or calcium. I promise there will be a reason for this, reasonings that you guys will find interesting. But G proteins are just special receptors or middlemen or relays between extracellular messengers and intracellular second messengers. So G proteins are kind of the middlemen to activate second messengers on the inside of the cell. You might be wondering, why are we learning so much about receptors? Different poisons, different drugs, different prescriptions all usually interact with these receptors on cell membranes. Because if a drug or a poison or tear gas or carbon monoxide can control some of these protein receptors, it can control what goes into the cell, and that will alter the function of the cell. So that's why we talk a lot about receptors, because different poisons, drugs, prescriptions, hallucinogens, LSD, marijuana, what they're doing is they're actually interacting with specific receptors on your cell membrane. So that's why it's important to just have a general idea of why receptors are important and what they'll do. Again, these steps take you through the G protein receptors, which are middlemen. So something will bind to a receptor, change a G protein, and the G protein will act indirectly on another protein receptor to change that signal that it eventually gets to the cell. And I'm gonna go through this because I won't expect you guys to know all these steps. And that takes us to the end of this chapter. Not the end of the chapter, the end of part B. There's still 2 more parts.